When monocytes exit the blood and enter the subendothelial space, they encounter growth factors which help them differentiate, known as colony stimulating factors. The macrophage colony stimulating factor, the macrophage granulocyte colony stimulating factor, and thus they then become larger cells which express different genes known as macrophages and dendritic cells. The situation is made a bit more complex given that there are different types of monocytes. There are different types of macrophage, and there are different types of dendritic cell. Macrophages, for example, can differ as to which T helper cells they interact with, whether they were pro or anti-inflammatory, and the cytokine local hormones which they produce. These different types of macrophages can also inhabit different types of plaques, whether stable versus unstable, and even different regions of plaques. Macrophages will more rapidly take up oxidized LDL particles rather than non-oxidized LDL particles, since the oxidized particles can bind to the scavenger receptors and are thus taken up more quickly. As macrophages take up lipids, such as the oxidized LDL particles, they begin to become the foam cells, the abnormal cells which actually promote atherosclerosis. They are irregular in the uptake of their cholesterol, in the combining of cholesterol with fatty acids at ester bonds, and they have a decreased efflux of cholesterol in that the ABCA pump, which pumps cholesterol into HDL particles, no longer functions as well. Thanks in part to the change in gene expression, which is mediated by oxidized lipids in the LDL particles, they then produce additional cytokine hormones, which can attract additional macrophages to the plaque. They can produce additional scavenger receptors, which will then increase the number of oxidized LDL particles that they can absorb. Eventually, they can undergo programmed cell death, and the byproducts of this can attract additional white blood cells to the plaque. Their increased size begins to occlude the lumen of the blood vessel, and they can contribute to the rupture of the plaque. So thus, these monocytes have become macrophages, which have become these abnormal foam cells, which are actually a primary cause of the negative effects of atherosclerosis.